Drug molecules interact with their receptors through a variety of chemical forces. Individually, these interactions vary greatly in strength, from weak van der Waals forces to much stronger covalent bonds. In most cases, drug binding does not depend on a single interaction. Instead, multiple interactions act together to determine how strongly and how selectively a drug binds to its target. We will examine each of these interactions in turn. Van der Waals forces are among the weakest interactions in drug receptor binding, but they happen everywhere across countless atoms at once. Even electrically neutral atoms aren't perfectly uniform. Their electrons are constantly moving, creating tiny momentary imbalances in charge. For an instant, one side becomes slightly negative, while the opposite side becomes slightly positive, forming a dipole. That temporary dipole can then influence a nearby atom, inducing a dipole there as well. When the charges align favourably, the atoms experience a weak attraction. Individually, these forces are tiny, but in large numbers, they help stabilise how a drug fits into its receptor, almost like molecular Velcro. But attraction only works up to a point. If atoms get too close, their electron clouds begin to overlap, and now repulsive forces rise dramatically, pushing the atoms apart. So van der Waals interactions depend critically on distance. Within a receptor binding pocket, some atoms of the drug will sit at ideal distances from nearby atoms in the receptor, allowing van der Waals attractions to form. Other atoms may be too far apart for any meaningful attraction, while atoms that come too close experience repulsion. At the optimal fit, attraction and repulsion balance each other, helping the drug sit snugly within the binding pocket. Hydrogen forms bonds with other atoms by sharing electrons. For example, in a water molecule, hydrogen shares electrons with oxygen. But the electrons are not shared equally. Oxygen, with 8 protons in its nucleus, attracts electrons more strongly than hydrogen, which has only 1 proton. As a result, the shared electrons spend more time near the oxygen atom. This creates a slightly negative region near oxygen, written as delta minus, and slightly positive regions near the hydrogen atoms, written as delta plus. An atom's ability to attract shared electrons is known as electronegativity. This animation illustrates the consequences of this uneven charge distribution. As water molecules move around, a hydrogen atom in one molecule may come close enough to the oxygen atom in a neighbouring molecule to experience this charge attraction. A weak bond then forms between the molecules. This is known as a hydrogen bond. Hydrogen bonds can also form between drug molecules and receptor proteins through interactions involving electronegative atoms. The atoms most commonly involved are oxygen and nitrogen. Fluorine can also form very strong hydrogen bonds because it is highly electronegative. Carbon and sulfur are much less electronegative than oxygen or nitrogen, so hydrogen atoms bonded to these atoms usually do not form strong hydrogen bonds. In general, the greater the difference in electronegativity from hydrogen, the stronger the uneven charge distribution and the greater the likelihood of hydrogen bonding. So, for a hydrogen bond to form, hydrogen must first be covalently bonded to a strongly electronegative atom, such as oxygen or nitrogen. The hydrogen can then interact with another nearby electronegative atom, forming a hydrogen bond. Here we can see two hydrogen bonds forming between a drug molecule and a receptor binding site. Although both are hydrogen bonds, in one the hydrogen atom is part of the drug molecule, while in the other it is part of the receptor. In either case, the electronegative atom to which the hydrogen belongs is called the hydrogen bond donor. It can be either in the drug or the receptor. The other electronegative atom contributing to the bond is called the hydrogen bond acceptor. So, in any hydrogen bond, one group donates the hydrogen atom involved in the interaction, while the other provides the electronegative atom that accepts the attraction. A single molecule can often act as both a hydrogen bond donor and a hydrogen bond acceptor at different positions, allowing multiple interactions to form within a binding site. Receptor binding pockets are formed from amino acids, and at physiological pH, many of their side chains exist in charged forms. For example, lysine becomes protonated and carries a positive charge, whereas phenylalanine remains uncharged at physiological pH. In contrast, aspartate contains a carboxyl group, which loses a proton and carries a negative charge at physiological pH. Many drug molecules are also charged. Acetylcholine provides a good example. It contains a quaternary ammonium group, which carries a permanent positive charge. As acetylcholine approaches the receptor, its positively charged ammonium group is attracted to the negatively charged carboxylate group of aspartate. This electrostatic attraction between opposite charges is known as an ionic interaction. Ionic interactions are generally stronger than hydrogen bonds because they involve full charges rather than partial charges. They often play an important role in contributing to strong and selective drug binding. In particular, they help position the molecule correctly within the binding site before other weaker interactions, such as hydrogen bonds and van der Waals interactions, contribute to the overall binding. Unlike van der Waals forces, hydrogen bonds, or ionic interactions, covalent bonds involve the direct sharing of electrons between atoms, creating a much stronger and longer lasting interaction. Most drug receptor interactions are reversible. The drug binds through weak intermolecular forces and can later dissociate from the receptor. Some drugs, however, contain reactive groups capable of forming long-lasting covalent bonds with amino acid side chains within the receptor. One such drug is osimertinib, a targeted therapy used in the treatment of non-small cell lung cancer, which acts as an inhibitor of the epidermal growth factor receptor. Osimertinib contains a reactive acrylamide group, capable of reacting with sulfhydryl groups in cysteine residues within the receptor binding pocket. As osimertinib enters the binding site, van der Waals forces and hydrogen bonds help position the drug within the pocket, aligning it correctly for covalent bond formation. The sulfur atom of a cysteine residue can then react with an electron-deficient carbon atom in the acrylamide group of osimertinib, forming a new covalent sulfur-carbon bond. This creates a stable EGFR osimertinib adduct. Such covalent attachment helps maintain prolonged receptor occupancy and contributes to sustained inhibition. As a result, binding is essentially irreversible. Covalent drugs do not always remain fully attached to their target. Instead, the drug can transfer a chemical group onto the receptor, permanently modifying the binding site. Aspirin provides a classic example of this. It acts as an irreversible inhibitor of the enzyme cyclooxygenase, which is involved in prostaglandin synthesis and inflammation. Within the active site of the enzyme is a serine residue containing a reactive hydroxyl group. The oxygen atom of this hydroxyl group contains lone pairs of electrons and can act as a nucleophile. The reactive part of aspirin is its acetyl group. Within this group, oxygen attracts electrons strongly, creating an uneven distribution of charge. As a result, the carbon atom becomes electron deficient and susceptible to attack by electron-rich atoms. Aspirin first binds within the active site through weak intermolecular interactions, which help position the molecule correctly for reaction. The serine hydroxyl group can then attack the acetyl group of aspirin, forming a covalent bond. During this process, the acetyl group is transferred onto the serine residue, while the remainder of the aspirin molecule is released as salicylic acid. The enzyme is therefore chemically modified by acetylation. As a result, inhibition persists until new enzyme molecules are synthesized. Binding is therefore functionally irreversible. This table summarizes how drug molecules bind to their targets through a variety of chemical interactions. Van der Waals forces arise from temporary fluctuations in electron distribution and are individually weak. However, large numbers of these interactions help a drug fit snugly within its binding site. Hydrogen bonds occur when a hydrogen atom bonded to an electronegative atom interacts with another nearby electronegative atom. These interactions are stronger and more directional than van der Waals forces. Ionic interactions occur between oppositely charged groups and are generally stronger than hydrogen bonds. They often play an important role in positioning drugs within a receptor binding pocket. Most drug-receptor interactions are reversible and rely on combinations of these non-covalent forces. In contrast, some drugs form covalent bonds with their targets through the direct sharing of electrons. The drug may remain attached to the receptor, or it may transfer a chemical group to the receptor, permanently modifying it. Covalent binding can be useful therapeutically, but because it permanently modifies proteins, selectivity is especially important to avoid unwanted reactions with other molecules in the body. Together, these interactions determine both the strength and the selectivity of drug binding.